welcome everyone. Good afternoon to everyone, and welcome back to another exciting discussion about sustainable farming. Today, we will be diving into the fascinating world of microbes. We will explore how understanding the intricate relationships within soil microbial communities can hasten to benefit farmers. So I'm Claudia. I'm the Academic Director of Food for Sustainability, which is a proud partner of GROW, an international consortium supported by EIT Food, aimed at developing capacity building programs to help farmers transition to more sustainable systems. It's really fantastic to see so many of you interested in microbes. And to that end, we have invited three distinguished speakers to share cutting-edge scientific advances and their practical implications for farmers. So I'm thrilled to introduce to you Michael Schloter from Technical University of Munich and Helmholtz Munich. Michael will be discussing how environmental changes affect microbial interactions. We then have the pleasure to have Tim Mauchline from Rothamsted Research, who will be presenting on the impact of fertilizers on beneficial microbes applied to wheat. And we also have the pleasure to have from Oxford University, Gail Preston, exploring the relationships between plant domestication and its influence on native microbiomes. I hope our audience will be inspired to learn more about these developments and particularly how we can make soil microbiology a powerful ally for farmers. And before we proceed, just a quick note to let you know that Food for Sustainability offers microbiome soil analysis for farmers using next generation sequencing. And what we do, we examine soil microbiome DNA to identify microbial communities that can assist farmers with biocontrol, stress adaptation, nutrient cycling, and bioremediation. So if you want to know a little bit more about what we do, we have here the email. So get in touch through Goedelab at foodforsustainability.org. There will be someone behind the email ready to answer your questions. And before we begin, let's just refresh our understanding of soil microbiology with some eye-opening facts. I think that Rita has prepared something for us. Right, Rita? So, yeah, fantastic. So we have a Slido. So Rita has prepared a Slido for us. So you can just use your QR code and mobile and use this QR code. You can scan and enter the Slido. Or if you want to use your PC, you just type in www.slido.com and in the hashtag box, just insert F4S Academy. And we can see that Rita is trying the QR code on her mobile to see if everything is working for you. I can also invite our speakers to take part in these very interesting questions. Rita, can we have the first question?
Sure.
Please. Okay. So the first question asks if you know what percentage of the world's species are found in soil, according to one of the most recent studies. Okay. Let's see. 64%, 59%, 40%, 25%. Everyone is voting. So we are, seems that our audience already knows that this percentage are quite high. How high are they? See if we have someone else voting. So we are talking about biodiversity in soil. So how many species are found in soil? So, and we have here a winner, 64%. some more votes for. Oh, yeah, it's really nice to see all these graphs moving and people voting. Great, fantastic. No one for the 25%. That's very interesting because an older study pointed that a quarter of all biodiversity on Earth was living in soil. Brigitte, can we have the correct answer? Yes, so a recent study from 2023 revealed this astonishing figure, highlighting the immense potential that lies literally beneath our feet. This is indeed an incredible number, and I dare to say that most of the common citizens are not aware that so much of our biodiversity lies in soil. So the most recent research points to 59% of world species, so the Earth's biodiversity living in soil. Thank you so much for taking part. Fantastic. Let's go to another question. Rita, what have you prepared for us? And this times we want to know how many bacterial cells are just in 1 single gram of soil. So we know that we have a lot of species living in the soil, but how many in 1 gram? I'm just talking about 1 gram, not 1 kilo, nor 1 ton, not 1 hectare. 1 gram of soil. Let's give us just a little bit more time, please. I don't want a tie. Thank you. That's more challenging with a tie. Fantastic. Okay, so we have our votes counted. So 1,000 bacteria, 100 bacteria, 1 million bacteria, 10 billion bacteria. Rita, 10 billion bacteria sounds like a very big number. Can you give us the correct answer? And it's indeed 10 billion. In just 1 gram of soil, there can be up to 10 billion prokaryotic organisms. And 10 billion is literally 1 with 10 zeros. Can you write 1 with 10 zeros? This is really a lot. The number of bacterial species range from several hundred to nearly 9,000. And this diversity, as we know, is crucial for many environmental processes that sustain all life on Earth. So this is really an impressive figure of what lies in just 1 gram of soil. And our speakers will tell us a little bit more about what's happening with these tiny organisms. Thank you so much for taking part, Rita. I think we have one more question. So finally, do you know which microorganism group dominates soil biomass when soil is undisturbed? Bacteria, fungi, nematodes, actinomycetes. Which group in terms of soil biomass dominates undisturbed soil? This is also an important part, okay? Fungi and bacteria very close, very tight. They are more fond of bacteria and disturbed soils. But I can tell that we have a knowledgeable audience with us. Thank you so much. Rita, is our audience correct? Undisturbed soil, soil biomass? Yes. Recently, research from the Swiss Federal Institute for Forest, Snow and Landscape Research showed that fungi take the lead with 90% of fungal species living in the soil. And they are followed by plants and their roots at 86%, while earthworms and molluscs like snails make up of 20% of the soil biomass. Thank you so much for taking part in this small icebreaker. So now we are into the topic. Fantastic. I'm now going to turn to our speakers. So I will start by introducing Michael Schloter. Michael is the leading microbiome researcher, focusing on the interactions between environmental microbiota and host microbes, including humans. Michael employs both omics approaches and traditional laboratory techniques to study microorganisms. After completing his PhD in 1994, he held various positions before becoming head of the Institute of Comparative Microbiome Analysis at Helmholtz-Zentrum in Munich in 2011. As of 2023, Michael is the professor of environmental microbiology at the Technical University of Munich. Michael, it's a pleasure to have you here. Thank you so much. The floor is now yours. We can see your presentation. Not in presentation mode. We are ready when you are.
Okay. Thank you so much for inviting me to this really very nice workshop this afternoon. It's a really big pleasure to tell you a little bit about the plant microbiome and its interaction with the soil microbiome. And I also want to stress a little bit why we are all so much fascinated by the plant microbiome. Because indeed, the plant microbiome is a key factor for plant health. You can see here a number of functional traits that are provided by the soil microbiome and by the plant-associated microbiome. And you can guess, without that traits, a plant is fairly impossible to live in a complex environment. So it's like things like nutrient mobilization, which is indeed very, very important, mainly in natural soils where no fertilizer is given. But there are also issues like control of phytopathogens. This biocontrol issue becomes more and more important also in sustainable agriculture because it can substitute pesticides if we know which organisms are responsible for these particular traits. But there are also traits like plant growth promotion, production of phytohormones, degradation of pollutants, of course, in the soil is a very important issue because it enables the plant to grow in the particular soil. And of course, the most known function is also the transport of water and nutrients by mycorrhiza fungi. So you can guess, maybe in vertical farming, you don't need microbes, but in any other natural environment, if a plant should grow, you need microbiota. And of course, when these microbiota are so important, the plant during evolution developed traits to recruit these particular microorganisms. And one very important trait is, of course, the root, because by the root, the plant is able to recruit microbes from the soil and take these microbes into their interaction with the particular plant surfaces, and then all these traits that I was showing you were happening. But of course, there's also above ground things happening. There also the plant is recruiting microbes from the air, and these microbes are also very important when we talk about UV protection, production of volatile organic carbon, and so on. So there are a lot of traits which are exclusively above ground. So this recruiting of microbes from the environment is a very, very important trait. But then the plant also has something like a self-security, which is the microbe which is in the seed and which is given from generation to generation. And mainly for the germination of the plant, these microbiota are indeed very, very important. And just recently, people discovered that the seed microbiota are indeed very important. And like for humans who transmit their microbiome from the mother to the baby, also there by the seed microbiota from the first generation to the next generation are transmitted. And this is becoming more and more important. But I would like to talk today a little bit about the soil microbiome and how the plant recruits the soil microbiome. And this is just an experiment we did with 50 different potato cultivars, and we grew them all in one particular soil. And we were curious if there are cultivar-specific differences in the recruitment. And we could group these different potato cultivars into different groups, some which yielded high yields, some which yielded medium yields, and some which yielded low yields. And then we looked on the microbiota, and we could see that each of these cultivars had its specific microbiome. And the challenge was to figure out now what is specific for these high yield potato cultivars, what is specific for the medium one, and what is specific for the low yield ones. And we could see that actinobacteria played a very important role. They were exclusively found by those cultivars which yielded higher yields compared to the others. There was also Marsilia, proteobacteria, which was very important and which was only found in those cultivars which has high yields. In contrast, there were some bacteria like acidobacteria, which were only found by these cultivars which yielded lower yields. And this indicates something, that the exudation, so the provision of the carbon by the plant, plays a very important role, and that there are really cultivar-specific differences. This recruitment of the acidobacteria indicated, for example, that there must be very low exudation rates for these cultivars, simply because acidobacteria are considered more as oligotrophs, and they are outcompeted by other microbes when there is high amounts of easy degradable carbon available. And this also is reflected by the actinobacteria and by the proteobacteria, which were dominating in these cultivars which yielded high yields. So the investment of the plant in exudates is indeed very important for this particular recruitment. And we could also show by a bioinformatic technique that this recruitment is not a process which is just randomly happening. We could see that most of the bacteria that we found for the different cultivars were really deterministic there. So there was a very specific process obviously going on, that these specific microbes were recruited by the plant, and therefore it is very important to understand how this interaction, how this recruitment process is taking place. We were asking then the question, is this microbial community at the plant-soil interface really important? So we stressed the soil and we autoclaved the soil to reduce diversity, and then we took a normal soil and this autoclaved soil, and we grew potatoes there, and we put a drought stress on the particular plants. And what we could see is, when there was no drought stress available, there was not much difference in the growth of the plants, independent if they had a normal microbiome in soil or a reduced microbial diversity. But at the time point where we applied the drought stress, we could clearly see that the cultivars were very much suffering, mainly in those soils where there was this reduced diversity of microbiota, meaning that under stress, the meaning of the microbiome becomes even more important than under regular conditions. And when we think about drought stress and climate change, we see that the microbiota become more and more important for sustainable growth of the plants. We could also see that there was different recruitment of bacteria. There were obviously some bacteria, mainly the actinobacteria, in the stressed soils with the low diversity missing. So they were substituted by other bacteria. But the substitution meant that obviously certain functional traits, mainly those traits related to drought resistance, were missing in the particular communities. So these plants were really very much suffering from the drought stress once they were in the soil with a reduced microbial diversity. And when we think now about what's happening at the moment, we see on all trophic levels losses of diversity. And we did a study, a meta-study, where we could also show that microbes in the soil are suffering by these biodiversity losses, and we could mainly identify land use intensity as a main driver for losses in biodiversity in soil. And again, we did a meta-analysis, and we could show exactly the same that we could see for the potato in this particular greenhouse experiment. because when the plant was growing under optimal condition, this reduced diversity in the soil did not matter much. But once there was acidification or drought stress going on, there was a significant reduction in yields, meaning that the diversity of the soil is really very important once there is a particular stress going on. And therefore, this reduction in diversity becomes a really big issue when we are talking about climate change. And when you ask farmers now in terms of potato, what they are really afraid of, and this is what they know from the practice, they all say drought and heat is the most important stressor where they are afraid of. And when you look on certain calculations and models, you can see that people are expecting losses from 80 to 30% in the upcoming years, just because there is this increased drought periods going on. So what can we do? One thing is we all love microbes because they just can develop to particular ecosystems. And just to show you the amazing capacity of microbes to evolve and to adapt to certain environmental conditions, here a study we did in the Atacama Desert, and you would not expect that there is any life, but then we could really identify microbes there that are living there, and they have very specific traits there, which enables them to survive in this particular environment. And they even transfer these traits to other microbes, which are transported there by air. And therefore, there's a really very complex community in an environment where you think there are no microbes available and no microbes can survive there. The problem, when you trust on this evolutionary pattern, this may take thousands of years. And this is, of course, something we do not want to wait. So let's think about other mitigation strategies. And one strategy, which I think is a very nice tool, is this so-called rhizosphere engineering. And Shilpi Sharma from India was the one who applied this, one of the first, in a very successful manner, because she was growing the plant with stress and she had different pots. And she always takes the plant and the soil from the pot, which has the biggest yields and where the plant was obviously not so much stressed by the stressors like in the other plants. Then he used the soil again for growing plants, and she always takes the best plant. And by that, she got a community, which was then really very, very helpful to support the plant and to reduce the stress and improve the stress tolerance of the particular plant. So this evolution, which I was showing you before, in this case, it is done in the lab, and it is much faster to get communities which are then helpful for the plant to mitigate a particular stress. And indeed, this has been applied now in many, many cases in a quite successful manner. And there are now a lot of communities available, which people also apply already, mainly for drought stress, successfully to enable the plant to mitigate this particular stress. Of course, we all have a lot of bacterial isolates in our freezers, and all of them have traits which support the plant growth. And it would make sense now simply not only to use one of these bacteria, but to combine different bacteria to bring different functional traits at the plant-soil interface to enable the plant not only to tolerate one stress, but maybe also to tolerate more stresses to improve nutrient uptake and. And therefore, this mixture of different bioinocula, which we call synthetic communities, becomes really a very important and very interesting issue. And just one example where we applied this successfully, this is now not potato, this is apple replant disease. We could identify a bacterium which was able to degrade one particular toxin produced by the plant itself in the soil. And we could identify also a number of other bacteria which together with this bacteria could enable the apple plant to grow better under these apple replant conditions. And we applied them together to soil, and we could really show that the apple plant was growing much better compared to without the inoculum, or even if we only applied one of these particular strains that we identified. So this concept of synthetic communities becomes very, very important. There's a last thing which I think is a fantastic tool, and this is the so-called soil transplants. And people working in medicine know this very well. When they treat Clostridium difficile infections in the gut, they give stool therapy. So they give the microbes of healthy people to compensate this Clostridium difficile infection. And the same can be done in soil. You take soil from a healthy environment close by and bring it to a degraded soil. And this indeed helps then to bring back functionality and soil quality to this particular soil. And indeed, people in Brazil, where they have large hectares of soil which are degraded by monoculture in the last 50 years, they apply their soils from cerradas close by just to compensate microbiota that had been lost during the last decades. And this works obviously pretty well. We also did this for recultivation purposes, where it is obvious that these soil inocula from a healthy soil play a very important role. We did it for open coal mining, where these areas had been recultivated after the coal mining had been stopped. And we could show that when you apply then soil from a healthy environment to this particular less material which was used to fill the gaps then, the regeneration of the soil was very fast. And even after 7 years, we were able to grow barley on this particular site. And the yields were not very much differing from a soil where barley has been grown ever since. So this inoculation strategy, which is somehow a black box strategy, obviously works very well. And just to sum up a little bit what I say, of course, microbes play a very important role, but they have to be embedded in a certain management context. They have to be embedded also in a certain cropping sequence. So monoculture is obviously not the best one when you want to promote microbial diversity. When we talk about intercropping and all that things, this is something where you really can induce microbial diversity in a particular soil. And of course, breeding becomes more and more important, because when we know now more about the substrates that the microbes would love to have in the rhizosphere, with genome editing and all these techniques, we are able now to generate plants which help then the microbes to survive at the plant-soil interface and do the particular functional traits. So it's not only microbiology, you have to implement the microbiology in the management, in the cropping sequence, in the use of the correct cultivars. And I think this is the big challenge which we are facing for the future, to make these use of microbiota in sustainable agriculture more successful. So with this, I'm through with my presentation. I would love to thank you all for listening. And of course, I'm very much open for questions. Thanks a lot.
Thank you so much, Tim. This was a very comprehensive overview of all the research that you've been doing, and all the things that you have been advancing in terms of science and knowledge. It's a really big challenge to squeeze all this in a 10-minute presentation, but that's fine. I know that we still have a couple of minutes here. Let me see if we can I will start with a question to clarify some of the topics that you have mentioned. For example, I was quite intrigued about your rhizosphere engineering techniques you briefly mentioned. So can you just explain a little bit more, how can we effectively implement rhizosphere engineering techniques to enhance crop resilience, namely against climate change induced stresses that you mentioned were some of the most concerns for farmers?
So what people are doing is they do pot experiments first, and they do this type of microevolution. So for 5 or 6 different rounds, they always select the pot where they have the best yield, so the best stress response of the particular plants. They take this up, and so they enrich these particular microbes, which enable then the plant to cope with the particular stress. After 5 or 6 rounds, these microbes, these microbial communities, are so much enriched that then people start to isolate them and to use them as particular synthetic communities, to apply them under field conditions when people are facing drought stress. This is how they are doing that at the moment. So this is, I think when you only are facing one stress, this is a very, very promising technique. But of course, the question is, in nature, you're often facing many different stresses together. And this is, of course, a challenge when you would do this evolution now for 5 or 6 different stressors. This is something what people are trying out at the moment. If then this approach also works very well and enables us to get the right microbial communities. At the moment, at least what we are doing, we are taking from our fridge the microbes which we have the genome sequences and where we know that they have particular traits, and then we mix them and see if it works out or not. Because even that is a challenge, because just think you mix a gram-negative bacterium with some fungi which is producing an antibiotic. In theory, both have important traits, but at the end, when you bring them as an inoculum to soil, they inhibit each other. And at the end, nothing is working. So even if you take microbes coming directly from the fridge, where you have particular traits, it's still a challenge because you don't know how these microbes interact with each other.
Yes, this cocktail that we still need to understand and see what comes out from this cocktail, but very promising. I'm really pleased to hear that you are also addressing that topic from the lab to the field. Michael, we have a couple of questions. Armando is asking, sorry, the question jumped here. What can be a cost-effective way for farmers to inoculate their soils, both at a small and at a large scale? This is coming from the field.
I can tell you about some experiences people make in Brazil, because this soil transplantation approach is so successful. Each of the large-scale farmers in Brazil has now hired a microbiologist. And what they are doing is they are not applying the soil directly. They are putting the soil from a rich environment to molasses, and then they take what comes out and put it on their field. And we have contact to one farmer, and he saves every year roughly $500,000 US for pesticides, just because he's applying microbes which are obviously doing the same job. So it is not a matter that this costs more. If you apply that in a very good manner, then you might even save costs. And to convince farmers, you always have to say, this might help you to save costs. And this approach, obviously, it's not proven now for long-term farming, but when you do it for 5 or 6 years, each year you save $500,000 US, so quite a reasonable amount of money.
Yes, indeed. Brazil is an interesting use case. Also poses some challenges to Europe, but they are definitely showing us some interesting results. And the cost side is very important, and they are achieving very impressive results with
maybe just to add that there is no misunderstanding. I would not advocate to take this approach one-to-one to Europe, because when you grow the soil bacteria in molasses, you get a lot of gram-negative, potentially pathogenic bacteria, also for human pathogenic bacteria, into your molasses, which you then apply to the soil. For them, it's a black box. They don't care. So they put it to the soil, and then they get the expected results that they want to have, and then, of course, they are happy. So in Europe, this would not be possible because we have very clear laws related to the use of bioinocular, and a black box approach like generating bacterial suspensions in molasses would not be possible. And I would also say this would be quite risky. So I would not say to transfer this one-to-one, but the idea to take microbes from a soil which is very good in quality and substitute the microbiome from a soil that is degenerated, I think this is still a very good idea.
Yeah, and the approach, it's a good way for us to start and try to understand more and to perfect the system. But of course, we need to have it more control. At least in Europe, we are obliged to have it more control. I will just speak 2 more questions, and I do apologize if I cannot have all we cannot have all the questions. Tima has a question here. He wants to know what type of technique you use to determine bacteria. Do you use Illumina or do you use nanopore?
So we, for barcoding, so just for making this phylogenetic trees and to assess diversity, We are using still Illumina, which is the working horse in our lab. We shifted now for the metagenomes and for the assembly of genomes completely to Oxford. We have the Promethean system in our group running very well now, and it works really very good. Sometimes we have to complement still with some Illumina data, but mainly for viruses and for plasmids, which we are very much interested in different contexts. This approach with the long reads works very well in our hand, and the chemistry of Oxford improved a lot in the last 2 years, and so the data that we get is really very much reliable.
Okay, very good. There's a question here wanting to know whether you have a study that shows that we are really assisting farmers to cut costs by using, microbiome farming. If you have, I would ask you kindly to drop here in the chat. I'm going to move on with our webinar. Michael, if you can just have a look at the chat and see if you can, just answer a couple of questions that we have here in the chat, while I call Tim Mauchline to the floor, and we need to move on. Time flies here. So let me introduce you to Tim Mauchline. He's a principal research scientist at Rothamsted Research, specializing in soil and plant microbiology. His research focuses on microbial interactions in the plant-root environment, particularly in wheat. Tim employs traditional and cutting-edge techniques to study the ecology of microbial communities in cropping systems. His work aims to understand the function of plant-associated microbiomes for nutrient cycling, stress tolerance, and disease suppression. Recently, he has investigated factors shaping the wheat root microbiome and its role in plant health, including the potential of specific microbes like fluorescence pseudomonas in protecting wheat against take-all disease. Tim, it's really a pleasure to have you here. I can see your presentation, not in presentation mode, but I'm sure you'll do that in a second. Thank you so much. It's perfect. The floor is now yours.
Great. Well, thank you very much for the invitation. It's great to be here this afternoon. So, and thank you for that the introduction as well. So, as you can see here by the title of this first slide, I'm interested in telling you about the possibility of microbiome facilitated wheat production. So as an outline of the talk, I just wanted to, I'm sure you all know quite a lot about this anyway, but I just wanted to give you a little bit of an outline of what the plant microbiome actually is, why we study it, and then what I want to do then is move on to 3 different case studies that we have of studying the wheat microbiome at Rothamsted. So that's to do with the impacts of soil fertilization. That's then to follow on looking at the impacts of wheat dwarfing, and then finally to combine those 2 factors to look at fertilization and dwarfing stroke domestication. So what is the plant microbiome? As I'm sure, as I say, we all know quite well, but just to recap really, we're thinking here about the different niches that can be occupied. So we look at the above ground parts, such as the phyllosphere. Then there's the below ground parts, so we're looking at the rhizosphere as well as the rhizoplane. In addition to that, of course, microbes can also colonize the plant internally, and these are termed the endophytes. And most of the microbes are, I would suggest, which are generally recruited or selected by the plant from the bulk soil, but of course there is vertical transmission as well. In terms of what microbes that we're interested in, well, it's all of them, but as we've already seen in the quiz earlier, there's a lot of importance of bacteria and fungi, but also things like protists and nematodes are also significant players. So why do we study it? Well, the soil microbiome has potential in contributing to more environmentally benign agriculture. It's well known that naturally occurring soil microbes influence plant health, growth, and resource use efficiency. So this can be in terms of nutrient solubilization or plant disease suppression, or abiotic stress tolerance. And there are well-known examples, especially things like rhizobia and arbuscular mycorrhizal fungi, but that we think there's a lot more range of microbes that we can explore further. And this has really been facilitated now by the recent advances in nucleic acid sequencing technologies. And this is now allowing us unprecedented ability to study these communities in much greater depth and resolution. This is in terms of the actual microbial communities at the community level, but also as genomes of individual microbes. So putting that all together, this then offers us a much better opportunity to understand and exploit this resource. So I suppose I want to just talk a little bit about the first part of the case studies, and this is to do with the impact of fertilization. So I want to introduce to you this one experimental field at Rothamsted where I work, and this is called broadbulb. It was established in 1843 as a long-term experiment. And broadbulb is able to explore many different factors. It's primarily concerned with wheat production. There's also different rotations present in it. And also there are certain plots with and without herbicides and pesticides. So it's a really nice resource to be able to study wheat production. And what we have decided to do is then start looking at the individual fertilizer treatments and how they impact the wheat rhizosphere microbiome. So you can see here we focused on 4 different treatments. That was the farmyard manure, that's what FYM stands for. There was the no inputs, so that's literally no input at all since 1843 into this particular plot. There's medium inorganic N and high inorganic N. And what we were able to see was that there was clear shifts in the microbial community in the rhizosphere as well as the bulk soil, but we're just showing the rhizosphere here, and how this was driven by the fertilization application. So you can clearly see that for these 4 different fertilizer treatments that there is very clear differentiation in the community structure. Just to stress that I'm purely looking at bacterial diversity in this study. So, in addition to that, I just wanted to then point out what actually are the shifts in these microbial communities in response to these different treatments. So what we found was that the actinobacteria and proteobacteria were always abundant, regardless of the nitrogen treatment. We found that the Bacteroidota increased in response to high inorganic nitrogen, and they're going to feature later also in this presentation in terms of their functions. We also found that the acidobacteria, the Lateski bacteria, and the planktomycetes were less competitive under high inorganic N. So, as you can see, we've had this functional, this description of the microbial communities. But what we wanted to do then was actually start assessing the microbial functions, and we did this in quite a sort of traditional way, I should say. So what we're able to do is make some microbial libraries of bacterial cells, which were obtained from wheat plants, which were either from a fertilized system or unfertilized system. And what we did is we assessed these microbial libraries for their ability to perform particular plant growth promoting functions. And we did this in terms of nitrogen solubilization, phosphate solubilization, phosphorus solubilization, potassium solubilization, iron and zinc as well. So you can see here that we've got a range of both macro and micronutrients. And if you look at these slides here, you can see where you have a non-fertilized situation, so that's the top panel, the microbes are much more active in their ability to actually show these beneficial functions for plants. So this is where, so when we're actually adding the fertilizer, this is suppressed. And so then to put this into a bit more context, if you can see on these graphs here, we can see the actual proportion of P, K, and iron solubilizers and non-fertilized and fertilized condition. And it's a clear response here that in the rhizosphere of non-fertilized plants have a much higher proportion of putatively beneficial microbes. And this is to the tune of 91% of the root bacteria in unfertilized soil having a beneficial trait, whereas only 19% of root bacteria in the fertilized soil having a beneficial trait. And just also this, the graphs on the left there are in terms of a proportion of solubilizers, but then actually if we combine all of those and look at the actual abundance, we still see that even in the unfertilized system, we have a higher abundance, not just a proportion of beneficial microbes. So what I want to do now is move on to the next part of the talk, which is to look at the impact of wheat breeding on the wheat, so on the wheat root microbiome. So by way of an introduction, and say that pre-Green the Green Revolution, stems were not strong enough to support the heavy grains, so plants fell over in a process known as lodging. And this resulted in increased risk of fungal infections, increased problems with harvesting, and as a consequence, decreased production and economic losses. So quite sensibly, the breeders changed the scope to actually have shorter wheat varieties which responded well to fertilizer application. But it should be said that that was done without really any consideration of impacts on what was going on with the microbiome, because this was obviously pre-microbiome days. So what we actually did in this study here was just to look at a range of tall and short hexaploid wheat cultivars. And what we could clearly see is that there was a difference in if you look at the PCOA on the left, you can clearly see that there are differences in the community structure with the talls and the semi-dwarfs, or the shorts as I call them, being separated. And then if you look at the richness and the diversity on the right-hand side, we can see there is also a split. And what we can see is that the dwarf cultivars have a greater richness and a higher diversity than the tall cultivars, which was a little bit of a surprise for us. So then, in terms of what the microbes actually are in these different systems, what we found was that there were clear differences in the tall and the semi-dwarfs. And what we're seeing is that in the tall cultivars, we were seeing a much greater proportion of proteobacteria, the Bacteroidota, and the actinobacteria. These are more abundant, differentially more abundant in tall cultivars. And interestingly, these are sort of phyla which we normally associate with plant growth promotion and beneficial traits. Whereas when we look at the semi-dwarf, we can actually see that the verruca microbia, the acidobacteria, and as well as the plantomycetes were differentially more abundant. And this is interesting as well, because these are microbes that we normally associate with bulk soil. So there's potentially something going on here, which is showing that there's been a reduced ability of the dwarf cultivars to actually select for beneficial microbes. So the third part I just wanted to move on to was the combination of these factors. So the first part being looking primarily at the impact of fertilizer on wheat microbiome. The second part being looking at the impact of plant breeding, in particular dwarfing, on the wheat microbiome structure. So what we've done here is we've actually combined those two factors in a large pot experiment. And so what we were able to do is get a range of different wheat accessions. So this goes for very ancient diploid wheats and diploid progenitor species. So wheat, of course, in modern day wheat is hexaploid. So in terms of the bread wheat, would be tetraploid. Oh, sorry, that's hexaploid. The pasta wheat would be tetraploid. And we also do have these diploids. In addition to that, we also have unplanted bulk soil. So what we could see here is that there are nice differences that we're able to detect in terms of the proportion of PGPRs, so that's the plant growth promoting rhizobacteria, in the different, according to the different ploidy level. And so what's very interesting is if we look at the unplanted soil on that graph on the left, that's quite similar, if you like, to the hexaploids on the right-hand side. But what we're actually seeing is that there are large increases in the proportion of beneficial microbes in the diploid wheats, in particular the B and the D types. But then also looking at the graph on the right, what we can see then is which bacterial phyla are being shifted in these different situations. And I mentioned earlier on about the importance of the Bacteroidota. So we know from the previous work that the Bacteroidota are always strongly abundant in response to high fertilizer. And that's also the case here. But what we're seeing here is that the actual Bacteroidota which are selected for in the presence of high fertilizer in modern wheats are not actually plant growth promoting rhizobacteria, but they are when we actually look at the diploid wheat accessions. So this is sort of pointing to the actual loss of beneficial Bacteroidota in modern wheats when they're subject to high levels of fertilizer, whereas this is not the case in the diploid wheats. So that's something that I think that we should consider for future studies. Okay, so I just wanted to say a few take-home messages, and this is really that mineral fertilizer application reduces selection of beneficial microbes in the root environment of modern wheats. And what we're also seeing is that wheat breeding, and in particular dwarfing and polyploidization, in combination with the mineral fertilizer application, has reduced selection of beneficial microbes in the rhizosphere. And this is, just to underline again, is primarily caused by a reduced amount of beneficial Bacteroidota in fertilized modern wheats. So when we take all of this, what the real challenge, I think, is to actually, how do we actually use this information to actually and apply it? So Michael in his talk was talking about the different ways that we can do this. And just to recap some of those things, really, we can think about inoculation strategies. And we could also think about selection of the microbiome, the indigenous microbiome by the plant. So, but I think really the way to, if we're going to really start thinking about selection of the indigenous microbiome, this would involve future development of wheat lines which are considering plant microbiome interactions. Okay, so I'd just like to acknowledge the team at Rothamsted, and thank you very much for your attention.
Thank you so much, Tim. It was really very, very interesting, fascinating work that you have there. And I know that you have a lot of other fascinating work, but we don't have time. But this was very interesting to understand why farmers sometimes complain that if they drop drastically their fertilizers, yields suffer a lot. And you just brought here some scientific evidence why this might be happening. So I have here just two questions to see if you could comment. What do you have any idea? What is the transition period from high rates of fertilizers to having a microbiome that has, built itself towards these beneficial bacteria you were mentioning? And also, I was also wondering while you were presenting about the generalization of your findings. So you mentioned broad bulk, so a very specific area, and you mentioned wheat. This is then on wheat. So can this be transferred to other areas, to other crops? What are your thoughts on it?
Yeah, okay. Well, if I understood correctly, the first question regarding levels, was this to do with levels of fertilizer? Is that what you were thinking? So
yes, when farmers are very dependent on chemical fertilizers, and when they drop those fertilizers, thinking that they the microbiology will do their job. That's why it's so drastic, because actually things are not there.
Yeah. No, I mean, I think that we're not going to get away from adding fertilizers to systems, but I think we can get a better balance is what I'm thinking. And as I mentioned at the end of the talk there, I think that if we actually can amend or select plants which are able to interact better with the plant microbiome, that's something that we can do. We're actually doing some work at Rothamsted at the moment where we've been looking for tipping points in fertilizer applications, and where we actually see where yield goes and where microbiome activity goes as well. So we're looking at that at the moment. We're studying we're doing this with modern wheats, but we're also interested in including the ancient wheats as well, to actually get a better understanding of how this works. And then of course the answer will somewhere be in the middle, I would imagine, because there's a reason we moved away from the tall wheats that fall over and get fungal infections. So but we think we can learn from those ancient wheats to make better modern wheats which are less dependent on sort of high levels of the mineral fertilizer. You also mentioned about Broadbook just a moment ago. So were you asking about other long-term experiments that are
No, I'm just trying to understand if we can transfer the findings to other settings, for example, which is in Portugal, the conditions are very different. For example, the south of Portugal. Can we apply the same rationale to other areas, eventually to other crops?
I think so. I think that okay, there are always extremes in conditions, but I think that the microbial functions that we're uncovering, I think that those are important, and I sort of really think that the actual it's what's important is that what the microbes are doing rather than who they are. And so if we then did a study in Portugal or anywhere in the world, we will find very clear differences in the microbiomes in terms of taxonomy. But I think that the functions that we're looking for in systems that are similar, so there will always be a certain amount of stretch in these things. That I suspect that in certain arid environments, some of the principles may be slightly different. But I think that I would hope that the actual application of the principles we've learned from a temperate system at this long-term experiment can be applied elsewhere, and also to different crops as well.
Okay, let's see if we find a project to bring you over to Portugal, because these are very interesting findings. Tim, we have a couple of questions from our audience, and while you were showing the bioassays about microbes that were under fertilization and not fertilized, the question from Armando is, at which part of plant's life were the samples taken to assess biology abundance?
Yeah, thank you for that. That's a great question. So that was all done at the flowering stage, and that's really important, because what we found is that if we're actually doing another experiment to really to really sort of put some solid results on this, but at the moment, anecdotally, we know that if you actually let the plants go too far and they become senescent, then the actual microbiome will break down, and it'll be the actual the diversity of the system increases. And I think this is just down to the whole degradation of senescing plant tissue. So we're studying at the flowering stage, which we think is a more ordered, mature community. But I should say we are also doing a study at the moment to go from seedling to the senescent stage, so we can actually really get an idea and a proper understanding of the microbial function over a complete life cycle.
Okay, interesting. Good, thank you. I will pick two more questions. I wonder if you can comment on this, Tim. Something to consider with the implementation of this knowledge and technology by culturing and adding microorganisms to the field. Are there some need to comply to regulatory requirements? We touched on this a moment ago with Michael, but are you aware how regulation might be supporting or restricting these sorts of techniques?
Yeah, I mean, I'm not an expert on that, so I don't really want to comment too much. But I think that Brexit hasn't helped things, I would say.
Okay. Okay, thank you. One final question from Gianpiero. Gianpiero, it's great to see you here. I have to say, very surprised and happy to see you here. Gianpiero has a question. Going back to what was said in the previous presentation, and this is a question, through genetic engineering, would it be possible to make plants, such as grasses, produce root exudates typical of legumes, for example, to promote the chemotaxis of nitrogen fixactors, thus reducing their nitrogen intake and avoiding excessive quantities of nitrogen fertilizers. We do have a technical question here, but Tim, any ideas?
I think that's a really tricky question. I mean, we know there are researchers. I think Phil Poole and Giles Oldroyd. They're really trying to develop nitrogen fixing cereals, so they'd be much better placed to answer that than I am. I do like the idea of altering exudates to attract microbes to the right place. But that's obviously that's very different to actually getting them to actually form a legume-like mutualism. But it's a nice idea to actually to think about how we can actually manipulate exudates to actually get them into the right place at the right time. I think that's key. But we may not need to actually go to legumes to do this. It could be just going back to ancient wheat cultivars to actually think about the exudates that they're producing, and can we actually get modern wheats to produce those?
Okay. Thank you, Tim. So food for thought. If you run out of
That's great.
ideas on your research, there's a suggestion from Gianpiero. Thank you so much. Tim, I invite you to just have a look at the chat, and there are a couple of questions. If you kindly have a look and spend some time here with our audience, I would appreciate. And I now call Gail to the floor. Let me introduce you to Gail Preston, that is a professor at the University of Oxford in the Department of Biology. And Gail specialized in molecular plant-microbe interactions. Her research focuses on plant-microbe and microbe-microbe interaction, with emphasis on bacterial diseases of plants and the mushroom microbiome. Gail aims to understand how microenvironments inside host organisms affect disease development and how environmental factors influence disease resistance. Gail employs cutting-edge techniques to study RNA binding proteins in plant microbe interactions and image plant pathogen interactions. Gail is currently the president of the British Society for Plant Pathology and the senior editor of Microbiology. Her work contributes to the development of sustainable agriculture practices and disease control strategies. Gail, it's a pleasure to have you here. So you can start sharing your
thank you very much. I should say, actually, I stopped being the president of the British Society for Plant Pathology about a month ago, so I need to make sure the website is updated.
Now we know. Thank you.
But I was for all of last year.
Okay.
So all right.
Thanks for the update, Gail.
Yeah, just a second. Let me see this coming up. Okay, can everyone see that? Okay. I'm just going to go to full screen.
Yes, full screen.
There we go. Right,
perfect.
I'm just going to do that. There we go. That'll make life easier. Okay, so I'm actually going to pick up a little bit where Tim left off and encourage you to think about how domestication and the changes in sort of processes associated with domestication might have affected the plant microbiota, and we can start then to think about how that might impact our sort of practices now. So, as I'm sure most of you are aware, the process of plant domestication, of taking a wild plant and creating a crop from it, has dramatic effects on what we call the phenotype of the plant, the characteristics or traits of the plant. So if you look, for example, at Phaseolus vulgaris, or bean, which is the crop we're mostly going to talk about today, if you go to sort of South and Mesoamerica, you find it as a vine, but modern sort of crops can often be grown in the field as you see, like a bush bean. This is a picture taken in South Africa of bush beans. And over the same time period, we see this change from these very small, wild sort of seeds through to the very big and differentially sort of coloured seeds that we might have today. And so what's happened over thousands of years is that domesticators have selected for plants that have the traits that they would like to have. So if we're looking at some of the more common things, often we're looking for yield, we're looking for a loss of seed shattering, so the seed stays on the crop rather than being dispersed to the environment. And we get changes in plant form and height, as Tim was talking about, changes in the flavour or the colour. Sometimes if we want to move plants around the world and grow them in different time zones, we need to think about changing their photoperiod so they'll flower at the right time of year. And we're thinking about getting them to have the cooking properties and processing properties that we might want. But at the same time, as we've started to sort of say domesticate plants, we do other things, or people have done other things without really thinking that this is a way in which they are shaping plants, but they are still shaping them in indirect ways. And so they're taking plants from the wild environments in which they would be found, they're putting them in agricultural environments, putting them through agricultural practices. Plants are exchanged by trade. We change their geographic distribution. We change what happens to them, particularly the seeds, once they're harvested. And of course, now they're subject to climate change, which is to say an indirect anthropogenic activity. And sort of not surprisingly, if we start changing all of these different attributes of plants, we're going to have an effect on the microbiome, which, as you've heard in the previous talks, is very much shaped by the plant and recruited to the plant. And so we could think that all of these different changes in trait are going to affect the microbiome all the way from the rhizosphere right up to the seed. And we heard in the first talk about all the different routes by which the microbiome is getting into the plant that are going to be shaped by these changes to our domesticated crops. Now, my colleague Kevin Foster here in Oxford wrote a paper in which he conceptualized the microbiome as sort of an ecosystem on a leash. So you can imagine that there is a connection between the plant and its microorganisms by which the plant influences the composition and the behavior of those microorganisms, and the microorganisms are also interacting with the plant and affecting the plant. And we might think that in a wild setting, these plants' micro interactions are subject to natural selection to favor best outcomes both for the plants and the microbes, that we might think that we're under selection for the plant to have a beneficial relationship with its microbiome and to have resistance to the microbes that it is encountering when a sort of disease is happening. Although at the same time, say, when we talk about pathogens, even in a wild setting, we have selection for some pathogens to cause disease. But when we start thinking about an agricultural ecosystem, we can think about that effectively creating a double leash where plants are being pulled in two different directions. On the one hand, we have the humans, the domesticators in this case, trying to get the plant to have the traits that they would like it to have. These traits such as yield or colour or palatability or cooking purposes or changes in seed dormancy. And in doing so, they're going to change the relationship of that plant with the microorganisms around it, because they're changing those traits that are acting on those microorganisms. And that might have consequences for the interactions those microorganisms have with the plant as well. And again, we've heard about that in previous talks about how that might have weakened the relationship between plants and beneficial microorganisms. So coming back to thinking about seeds and fruits, which is going to be the main topic of this short presentation, as we've seen, they're subject to really major phenotypic changes in size and in content. If we think about wheat, we can think about the fact that we select for wheats that have very different grain types. That goes along with changes, in one of the antimicrobial proteins that's in the wheat grain, or changes in the level of gluten to get different processing. So we're changing the environment that a microorganism would encounter inside the seed, and we're changing the processes by which that microorganism might get to the seed. So the seed microbiome becomes a very interesting test case to think about how domestication has actually shaped a plant's relationship with its microbiome. And the seed is also one of the examples of sort of a plant compartment where we see very big differences in what happens to a seed in a wild system compared to an agricultural system. In a wild system, some seeds might go through an animal before they reach the ground. In a bean, they're going to be sort of flicked straight away from the plant, and they're going to go straight into the soil. Their environment they're in between being dispersed and germinating is going to be very different from a domesticated situation where we're going to take our beans, we're going to dry them out, we're going to thresh them and mix them all together. In some cases, in sort of modern seed production, we really get them down to a level of dryness they would never have in a wild setting. And then we store them for really extended periods of time before we then germinate them. And during all of this time, the microbiome is changing, and particularly if we start applying other treatments like seed treatments and heat and chemicals. So by the time that seed reaches the soil, its microbiome has sort of say experienced a very different seed and very different processes from what we see in a wild plant. And as we've seen, domestication is also affecting the transmission of microbes, to and by the seeds. As you've heard in the previous talks, the plants are largely picking up their microbiome from the environment, although some of it comes from the seed they start with. But all of these changes influence that passage of microorganisms to the seed. As they get into the seed, they're probably finding seeds that have a very different chemical composition to the wild seeds, that they might have sort of say more defensive or secondary metabolites or specialized metabolites in the wild seeds, perhaps a more emphasis on just basic sort of carbon and nitrogen sources in the domesticated seeds. So less defensive, sort of say more nutrients might be sort of one of the balances that we get. So a very talented student in my group, Ricardo Saladin, started thinking about this, and he realized that Phaseolus is a really useful model system to think about what happened when we've domesticated the seed microbiome. As we've seen, the plants themselves have gone through big changes, and so have the seeds. But not only that, one of the big challenges of studying domestication is if we really want to do it, we need to travel in time. And beans are one of the very distinctive crops that were actually domesticated more than once. They were domesticated in the Andes and they were domesticated in Mesoamerica. So we actually have a replicated experiment where we can say they were domesticated twice, what happened in these two different replicated events. And also they're one of the crops where we still have the wild relatives available. There are other crops, including legume crops, where we've already lost the wild equivalent. We don't even have it to go to sort of anymore. So we've got the wild relatives, and we have this replicated event from about sort of 8,000 years ago in the Mesoamerica and the Andes. And so we can then start to say, well, were there consistent phenotypic changes other than just size through these domestication events? So one of the things we looked at was mineral sort of content. And so if we look, for example, at calcium, we can see that there is a higher proportion of calcium in the wild seeds compared to the domesticated seeds. And we could think about, why this might be. Where is calcium in a seed and what does it do? This actually isn't a bean seed, but it shows you what I want to show you, which is that the calcium, which you can see here in red, is in the seed coat. So it's on the particularly enriched. So it's throughout the seed, but it's particularly enriched in the coat, that sort of hard coat around the seed, and it contributes to the hardness of that coat. This was made using a technique that we use in my laboratory where we can actually study the distribution of minerals inside plants. You can see that the embryo inside the seed comes up in white because of its high iron content. And so why do we have these changes in seed coat calcium? Well, for Phaseolus, we know that seed coat calcium is associated with the hardness of the coat. And if you reduce it, then you get beans that actually are easier to cook. They soften more easily. And so it's logical to think that when people were domesticated beans, they wanted to sort of select for beans that would be easy to cook. You wouldn't get some hard, crunchy beans, you get nice soft beans. And so you select for reduced seed coat calcium along with bigger seeds. So we're getting these changes in the chemistry of the seeds that also potentially going to have impacts on the microbiome. This is just one of probably many changes in seed chemistry that have occurred. So Ricardo wanted to set up an experiment to really ask, what changes did domestication do to what did it implement for the seed microbiome? So he went to the seed bank in Colombia, and he got hold of representative replicates of these the wild populations and their domesticated descendants, and we carefully picked out the ones that seemed to be representative of the ones that had descended directly rather than blended too much over time. And then we grew them in a greenhouse experiment, all in the same soil, to try and give them the same starter microbiome. And then when the pods were ripened, then we took the seeds straight out the pods. We didn't mix everything together. We just took the seeds straight out the pod. And those are the seeds that we used for our microbiome analysis. And we thought we'd see something subtle, but in fact, we saw something rather dramatic. So we found that in this greenhouse experiment, our domesticated seeds seem to have a much greater proportion of proteobacteria, and particularly Pseudomonaceae, than the wild seeds. And this is more dramatic than perhaps had been previously reported because, as I've mentioned, seeds normally go through a drying process before they then go on to further analysis. In this case, we'd gone for a relatively fresh seed, and that drying process does tend to reduce the sodium Pseudomonaceae over time. But you can see these fresh seeds that the implications are dramatic for the difference between the wild and the domesticated seeds. And the other thing that struck us is that when we looked at the wild bean seeds, it was only in the wild bean seeds that we actually found rhizobia, these nitrogen-fixing bacteria that we've talked about earlier, in the seed. And rhizobia, we normally think about it coming to the plant through the soil, the roots, but there is possibly a route through the seed that might be happening in the wild plants, but not happening in the domesticated plants to the same degree. We're able to see that there were some specific microorganisms that did seem to be different between the wild and the domesticated plants. So just two particularly came up through the greenhouse experiment with Phaseolus vulgaris. And again, if we're thinking about why this might be important, why would we talked about proteobacteria as beneficial group, but it's also a group that includes a lot of plant pathogens, including the pathogens we study in my group. I particularly study a group of pathogens called Pseudomonas, Pseudomonas syringae and Pseudomonas sevastani, which causes a disease called halo blight of bean. And so if you're making seeds in which proteobacteria and Pseudomonas do better, perhaps you are creating seeds in which Pseudomonas pathogens are going to do better. And here you can kind of see the Pseudomonas causing their symptoms on the bean pods. You'll see another method we use in our lab, which is to actually make our bacteria glow. We make them bioluminescent, so we can see where they are as they're colonizing, in this case, the bean pods and seeds, and track their movement through the plant. So when we looked holistically at this data, what we see is that in the wild plants, we see to a certain degree quite distinctive populations, more separation of populations. In the domesticated plants, we see that their traits start to get more similar. In both domestication events, people were selecting for similar traits. So perhaps understandably, the bacterial population also starts to get more similar than when we compare the wild population, or perhaps in both cases, there is reduced selection to a certain degree on the microbiome. You can interpret this in a few different ways. So looking to the future, I think it's really important that we start to think about how plant domestication has affected plant health. We can see this model that in a natural ecosystem, we might have more natural selection for beneficial seed micro interactions that affect the germination and the stress tolerance of the plant, that might provide better protection against pathogens. And we might currently be in a situation in agricultural situations with domesticated crops where there has over time been reduced selection for these beneficial interactions, In the process, we're just selecting for the microorganisms that are best at colonizing the domesticated crops. Maybe we've got a situation where the plants are more permissive, for example, for seed-borne pathogens. We have perhaps differences in changes in the transmission of the seed-borne microbes, and we can think about the consequences of what we do to our seeds in shaping the microbiome that they start off with when we put them in the soil. So hopefully that's given you a bit to think about, and I'm happy to take any questions.
Gail, thank you so much for this presentation. Very interesting, very relevant also, and also congratulations on your research and your research center. So you touched two very interesting topics. So we talked about when we domesticate, we have implications in terms of nutrition, and this is really aligned with a very recent study that showed that our food is less nutritious than it was a couple of decades ago, and you just unveil part of the reason why it be. And also, you also mentioned very clearly the implications that this domestication has on microbiome. You had here the visual of the leash. You taking microbiome by the leash. My question is, we know that it has an influence. So plant domestication and the way we manipulate our plants has an impact on microbiome. Are we aware of those implications, or we are just learning after all this manipulation? So basically, when we do this domestication, are we really aware of the consequences that is happening on the microbiome, specific consequences, or we're just learning afterwards? You are muted, Gail.
Now, I think obviously the individuals who were domesticating plants, a lot of these events are thousands of years ago, so they were not thinking about the microbiome of their plants, except perhaps in some fermentation sort of context. They were just selecting for plants that had the traits they want, that seemed to survive in the field, so they're probably still selecting for an element of disease resistance in those plants. So this focus on thinking about the microbiome is still a relatively new part of our sort of thinking holistically about the plant and the microorganism. And it's driving, things that are really important, like becoming aware of how important it is, to, for example, to preserve the wild relatives of crops that have this diversity, not just in their genetic makeup, but also in their, their relationship with the microorganisms that they interact with, so we can better understand that. As I said, we're already starting to lose some of the wild relatives, so that becomes a really important priority for the future.
Yeah, that's also interesting, so that we don't use all that genetic diversity and all this patrimony that nature gave us, and we are just using it so selectively, and sometimes we're not really aware of all the consequences. Gail, just a clarifying question from Armando. He asks if this is right. Wild seeds promote a larger diversity of microorganisms independent of the growing medium.
I think it's difficult to sort of literally say diversity. I think we can say that the wild ones sort of say are definitely have a different diversity, but they're also shaped by the growing medium. So it's sort of it's a combination of factors acting on them. I suppose the way I think about it, and whether this is true or not, I don't know, is that these changes, these loss of defensive metabolites, perhaps have made the seeds more permissive to colonization by certain competitive, abundant organisms in the environment, and perhaps some of the organisms that could only colonize the seed in a wild plant because they can tolerate antimicrobial compounds present in those seeds. That's the sort of the difference in sort of diversity that we're seeing. They may be outcompeted more by these sort of, say, in the domesticated seeds by other organisms, or losing some ability of the plant to kind of proactively select for those.
Okay, interesting. Do you have data on microorganisms diversity regarding GMOs versus natural ecosystems?
So I haven't specifically looked into that side of things, but obviously, if you start genetically modifying organisms again, that's one trait among many that you're going to change, but it will inevitably change the microbiome. It's something I'm very interested in because we think a lot about minerals in plants. We think about zinc and iron and calcium. There's a push to do research in what's called biofortification, which is increasing the mineral content of the plants that we eat, sort of having tomatoes with more zinc and beans with more iron. And I'm very interested in thinking, when we do that, when we engineer those plants to have more minerals, what does that do to the microbiome? Is it good? Is it bad? Is it just, it's going to change, but what are the consequences of that?
Yes, at Food for Sustainability, we are trying to make the causal relationship between having more sustainable farming practices and more food nutrient density, which this is something that it's quite intuitive but hard to prove. So we have ongoing projects that just look into that. So not the domesticating, just through farming practices, trying to understand the impact that these have on food nutrient density, because instead of having to eat 4 apples, I would say the humanity would be better if we could have 1 apple and we got all the nutrients that we need. Okay, Gail, thanks very much. I have here one just final round question because time flies, for you and for Tim and Michael. And the question is, and I come from, a management background. I am always interested in these questions. What do you think is the biggest challenge in translating microbiome research into practical applications for farmers? And in your view, how do we think we can overcome some of these challenges?
I think I might let my colleagues who are closer to the coal face of this sort of take that question first. Tim, do you want to?
Yeah, sure. I think that for me, the main challenge is, I think I mentioned at the end of my talk, that there's 2 ways to go, really. I think you can either inoculate or you can recruit from the actual indigenous microbiome. And I really think that the way to go for a robust sort of microbiome facilitated agriculture is the actual recruitment, and that's going to be driven by the plant itself. So it's really going to take either some CRISPR modification or selective breeding or whatever it is, but essentially to make plants actually be able to select or recruit microbes. I think that actually the inoculation of microbes can work, but I think that there's quite often the sort of issue of, I've heard it once before in a talk, I think it's by Stephen Rolfe mentioned it, something about the sheep in the Serengeti. If you can put microbes in a particular environment, but they'll only do so well for so long until they're outcompeted by the indigenous microflora. So that's for me the big challenge, and I think that's the way to get around it, is to better understand what we can learn from the wild and undomesticated accessions, and those genetic components, can they be transferred to the modern cultivars to get the best of both worlds?
Yeah, no, I totally support that. You do see, a lot of people think, oh, if we go to these wild plants, we can get these magical microorganisms off them that will be so much better than the micro. And unfortunately, that's unlikely to work, because the domesticated and modern cultivars don't have the right traits to recruit them. They're not necessarily going to be competitive. So thinking about how we actually make sure that the soil composition has the microorganisms in it that are beneficial, that can be recruited by the plant, and then we think about getting the plant in sort of be able to recruit those beneficial organisms without while still being an attractive crop. So
Yes, and I would just add that probably also transferring part of this knowledge to farmers and to consultants, and trying to understand what are the implications of every single management that we do at the farm level, what are the implications, and how we can manage all those decisions to ensure that we are working to promote more beneficial microbes and bringing them closer to plants so that they can interact as they like to do on nature. I will need to thank you and pause here and stop these discussions. Apologies, but our time, runs very, very fast. I think we had unveiled how interesting and what sorts of things research is moving forward. So thank you so much for being here. It was really, really interesting. Hopefully we'll be in touch. It was really interesting to hear your thoughts. For our audience, just before you go, we have prepared a very, very short questionnaire based on what was discussed here. This will take you roughly 5 minutes. We invite you just to jump on these forms. We can give you a certificate if you take part on these questions for your presence here, and to ensure that you were here and hear about and interact with us. So Rita, we already have here, so you have the QR code again, or we also have the forms on no, on our chat. Rita, would you like to share, or is this fine, and just let everyone just at their own pace? So basically 3 questions, very simple questions. Don't worry, we won't be here seeing your answers, but the first one is which of the following is identified as the biggest threat to potato production in Europe? Michael mentioned this. Remember, there was a graph, and I know this depends on the part of Europe that you are, but specifically the results that were mentioned here you remember soil erosion, pest infectants, climate change, and water scarcity. The second question, team addressed the second question. What effects does the use of inorganic nitrogen fertilizer have on microbial communities in wheat soil? Tim, will be very, very sorry if anyone misses this question, right? Does it increase microbial diversity? Does it decrease? No effect, or it makes the microbes stronger? I think this is also easy, and we discussed this quite a lot. And then Gail mentioned about domestication. How does domestication affect the relationship between plants and then microbial communities? We've just discussed this. Domestication strengthens the plant relationships with their microbes. Domestication has no impacts. Otherwise, Gail will not be here. Domestication leads to a double leash, where both humans and plants influence the microbiota. Domestication eliminates microbial communities in plants. Okay, we can leave you. This is just this, and then we can go back to you with a certification if you wish. Thank you, guys. I think it's now 3 minutes past our hour. Team and Gail are British, so punctuality is something that it's it's how do you say, it's something that we always identified with the UK. But thank you so much. It was a pleasure. We will continue next Thursday, same time, same hours, same link. We'll be here this time talking about how farmers use soil microbiome, what they are currently doing, and what are the purpose and the results. So I hope you can join us next Thursday at the same time. Thank you very much to the audience. A special thanks to the speakers, and I hope to see you soon. Thank you. Bye-bye.
Bye-bye.
Thank you all. Bye bye. See you next week.